Hello, biologists. Welcome to lecture. Today we're going to discuss animal general body plans and what makes an animal an animal. So I have a picture of my two favorite animals right here, Parker and Max. All animals are going to be multicellular. If they're unicellular, they're usually put into a different group. Think about protists. Most are going to be heterotrophs. Hetero means different, and troph is eating. So heterotrophs have to eat their food. They can't make their own food. If you can make your own food, you're called an autotroph, so self-eating or feeding. And that would be things like plants that can do photosynthesis. They can feed themselves with photosynthesis. Animals cannot. They have to eat somehow. Animals do not have cell walls, unlike plants and fungi, and animals have specialized tissue. Some don't, but most do. If you're curious about specialized tissue, think about muscles and neurons, and we'll talk about it in a second. And animals also have a development of a body plan, which is guided by conserved genes, specifically the Hox gene, which we're about to talk about. Conserved means that they've been reused over evolutionary time. So it's something that was developed earlier and is kept. So it's conserved. We estimate that there is 3 to 30 million different species of animals on Earth, which is a really big range, but we're discovering new animals constantly, so we don't know for sure how many there are. Most animals are going to follow this pattern of development. You're going to start with two gametes. They're going to fuse. Your diploid gamete or your diploid zygote is from the fusion of two haploid gametes. That zygote is a single cell. It has all the genetic information needed to make whatever that critter is. It's going to do mitosis. So one cell turns into two cells. Then two cells turn into four cells. Then four cells turn into eight cells. And that's where we get our 8-cell stage. We're going to continue to do mitosis, which is referred to as cleavage because you're cleaving one cell into two until you form a little hollow ball. This is called our blastula. Note that between the 1-cell, 8-cell, and blastula stage, our grouping of cells hasn't gotten bigger. The cells themselves have gotten smaller. And then in my little blastula, think of it as like an inflated balloon that's kind of limp. And then you can push on one part of the balloon and the membrane of the balloon will fold inward. That's what we're going to have happen. We call it gastrulation. And the little pocket that folds inward, that's called the blastopore. So this is our gastrula, and the fate of this blastopore will determine whether or not something is called a deuterostome or a protostome. That's really important classification, and we'll see that on a phylogenetic tree coming up. Our complex tissues that we mentioned, we have four main ones. All of these tissues are going to be connected to each other on a cellular level with something called gap junctions. Gap junctions are like pieces of the plasma membrane that have little holes that so they can exchange things through, and then they're stitched together so they're really adhered well. And there are different types of cellular junctions, but gap junctions are really common in animal tissue. We're going to have connective tissue. Connective tissue is tissue that's embedded in an extracellular matrix. So extracellular means outside the cell, and matrix is the thing surrounding what you're talking about. So I'm sitting in my office right now. I'm surrounded by air. That's my matrix. If I was in a freshwater pond, the water surrounding me would be my matrix. If I'm thinking about the cells that line my capillaries, I will have extracellular fluid outside them, and then I'll have my blood inside them. So that would be the matrix that they're sitting in. It's whatever's surrounding your cells. We're also going to have epithelia cells. Epithelia cells line, they cover, they protect, they separate. Think about your skin. This is epithelium, but you also have epithelium going down your entire digestive tract, through your respiratory tract, covering your eyeballs, lining all your viscera, so all around your organs. It's going to protect everything and make sure that when you move your body. You're not creating friction internally. So all of that really important. We're going to have nervous tissue. That's our third type. Nervous tissue is used to send electrical impulses. So that allows me to sense pressure, to sense pain, to sense temperature, to have thoughts and feelings, but also to take those thoughts and feelings and turn them into locomotion. So all of my movements, my heart beating, my stomach gurgling, all of that is controlled by neurons that are integrating my sensory information from the outside and turning it inward. So it's really complex. We're going to talk about those cells in detail later in the term. They are gorgeous. The way that they work is so cool. I'm really excited for you to learn about it. And our last specialized type of tissue is muscle. So muscle, think about movement, locomotion, so your ability to walk upstairs or dance, but it's also more than that. So I have little tiny muscles in my eyeballs that control how much light I let into my eyeball. My heart is a muscle. My lungs are controlled by different muscles. My intestines have circular muscles that smush food through them. All of that is muscular as well. So it's not just our voluntary skeletal muscles, which we often think about. It's all of our subconsciously controlled organal muscles as well. So we have three types of muscles, and we'll get into that soon. Now, we talked about those conserved genes briefly. Conserved being they're used for a long time evolutionarily because they're beneficial, and so they have been kept. They've been conserved. The example we love are Hox genes. So this is a shortened of homeotic, Hox. And Hox genes control body patterns of where different pieces of your body develop. We think that duplication of these Hox genes allowed us to go from something that didn't have a body plan, like an amoeba, to something that had a more complex body plan, continuing segments like a worm, to something with a really complex body plan, like us. Our arms are different than our legs, which are different than our torso. All of that development is controlled genetically. We think that all vertebrates have this, because we know that there are 4 types of Hox genes, and so far every vertebrate we found has at least one copy of each of those 4 types. Lots of them have many copies, but at least one is found in every vertebrate. Here's an example. I have Hox A, B, C, D, so those are 4 types of Hox genes. I have numbers of them up to 13. And then these are color-coded to show you which part of the body they correlate with. So these Hox genes that are 1, 2, 3, 4, they're going to be controlling the development of cephalization, which is a head with sensory organs. Here I have a mouse embryo. You can see those colors are correlated with the head part of the mouse embryo as well. Then we're going to have the neck region. That's the next chunk. We're going to have the torso region. That's the next chunk. Each of those controlled by these genes here. And then we're going to have extremities, our last set of Hox genes that control the development of arms and legs. And that is true for your mouse embryo as well. Now, if you think of these as like programs, each program can be repeated. So each band in these organisms represents a different segment of a body. This, what is this, a brown-purple color? This brown-purple color is controlling a head. So all of these organisms have a head in the front denoted by that color. Then we're going to have a orangey thorax. Here's your thorax. We're going to have the orangey limb area. Here's the orangey limb area. Then we're going to have this light yellow abdomen. So here's some things with some light yellow abdomen. And then at the end, if you have a specialized butt region, that would be denoted by this light yellow color. So if I think of each of these as a genetic program, for something like a millipede, I would reuse the program for legs, right? So I'd use the head, I'd use the thorax, and then I'd say leg, 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 leg. And that would be repeats of these Hox genes. So once you have the genetic code for it, you can cut and paste and reuse. And so your millipede is running that code for legs several times until it gets to the butt. Same for the centipede, but theirs is more thorax instead of leg. Then we get to like a grasshopper. Each of those segments is really conserved. The wings attach here, as opposed to a bee where the wings attach right after the short little part of the head. So it helps us explain the body forms based on the Hox gene variation. Now we can also mutate Hox genes. If we take a Hox gene and we know that it is used for the creation of a leg, and we put it somewhere where it shouldn't go, like where this antenna is, your critter will develop a leg in the area where it used to have an antenna. And so we've done these experiments on fruit flies and bees and flies, and we can create little monsters like this by cutting the leg gene, the Hox gene, and putting it in the antenna region. Here we have a fly. Flies have this little balancing structure called a haltere. This haltere, like, it's thinking about, when your cat runs and it swings its tail back and forth. That's what these do for flies, is they help counterbalance your fly while it's flying. Now, if we remove this and we insert the gene for wings, we can create a fly that has basically two thoraxes with a double set of wings and no halter. And so it's experiments like these where we copy and paste the genes and see what happens to the body form that really helped us understand what Hox genes do and how important they are to body development. Let's look at this phylogenetic tree real quick. All of this here is animals. We're going to call them metazoa. Think zoa for animal. Then we're going to divide our animals based on whether or not they have tissues. The only guys that don't have tissues really are sponges. Everybody here in the parahoxzoa, think hox for body form, they're going to have tissues. So hoxzoa, that body form animal. Para as a prefix means like almost, like it's paranormal. It's not quite normal. So these do not have the hox. They're almost tissue containing. That's going to be our porifera, our sponges. So we also divide our organisms based on body plan. We can determine body plan by looking at symmetry. If you are bilaterally symmetry, you have a dividing line and your right and your left halves look pretty similar. We're bilaterally symmetrical. If you divide my right and my left side, both of them are going to have an eyeball, both are going to have an arm and a leg. So it's pretty much the same on each side. But note that you can only divide me from right to left. You cannot divide me from front to back. I'm only bilaterally symmetrical in one plane. That's true for this beetle as well. You could only divide it right to left, not top to bottom or front to back. Now, other organisms in the animal kingdom are going to have radial symmetry. For example, this is a coral polyp, or it could be a sea anemone, whatever it is. Could be a jellyfish. If you look at it, I could divide it on this plane and the front and the back are the same, or right to left and the front and the back are the same, or at a 45-degree angle and the right and the left 45-degree angle sides are all the same. So it's dividable in many different directions, hence radial symmetry. And then we have our weirdos, our sponges, the porifera. They don't have any symmetry, so they are not symmetrical. We have another weirdo that's not on this little diagram. That is sea stars. I always want to call them starfish, but they're not fish. Sea stars. They are radial symmetry, because if you go down each of their arms, they'll divide in half, but it's not bilateral because it's not only one plane, it's in 5 different planes, so pentalateral. We discussed the types of tissues. We have those 4 main tissues that helps us compare our different organisms and classify them. We mentioned those germ layers, but we're going to get into it a little bit more. There's some more we need to talk about that. And then we're going to discuss body cavities. We know that Hox genes control body plan, but the body cavities also help us classify animals. So if you have radial symmetry, this is really good for sensing and feeding all around you. So here I have a sea anemone. It doesn't have a front and a back. It doesn't have a forward. It just is everywhere above it is fair game for this little mouth to digest everything. This is common in drifting organisms, such as jellyfish, or sessile organisms, which means they're not moving. This little sea anemone, it can swim. I've got a video of that, but it's going to be attached to substrate, so it is spending its life non-modal. We call that sessile. This is often paired with simpler cephalization. So this has a mouth/butt. It has those feeding tentacles, but it doesn't have a full-on head with sensory organs. In comparison, bilateral symmetry is really good for directional movement. So if I look at my tiger here, that tiger is forward facing. It is hunting prey in this direction. It will attack in an anterior motion. So that's bilateral symmetry is really good for sensing the world in one direction at a time. It's usually paired with cephalization, which means having a head, and complex senses such as sight. This cat has amazing eyesight, hearing, smell. Maybe it's using that little pheromone taster in its mouth. This little cat here is going to be sensing a lot of the world and responding to it with its nervous tissue and its muscular tissue. This also supports complex body systems where we divide front to back and right to left. And we're going to talk about that with body plans. Here's bilateral symmetry in more detail. So we did say that we are the same symmetrically right to left, but not top to bottom. We have words to describe each of these planes. So my right to left plane is the sagittal plane. My top to bottom is the transverse plane. And my front to back is the coronal plane. Coronal means crown or head. So think we got front and the back of the crown. That helps me remember coronal. If you're talking about things that are more up anterior in the head, we call that cranial because it goes towards your cranium. And if it's down, we call it caudal. Caudal actually refers to tail. We don't have one, but if we had one, our tail would hang down by our feet. So that helps me remember it. Now here I have my torso. If things on my extremities are closer to my torso, I say that it's proximal. If they're further from my torso, I say they're distal. For example, my elbow is proximal than my wrist, which is distal. So this is further from my torso than my elbow is. So that helps you figure out how far down the extremity you go. In the front is ventral. We also use anterior. And in the back is dorsal. We're going to talk about this when we talk about fins. They have dorsal fins on their back. That's also posterior. Then we have toward the midline or away from the midline. So my nose is medial. My eyes are more lateral. My ears are even more lateral. They're further from the midline going down. All right. Let's look at our tree. We've added a couple traits. We know that we have specialized tissues in our animals. Well, everyone does, except for if this is on this branch here, that means that it's from here forward. That leaves out whoever is on the other side of this branch. These critters, the Placozoa, they do not have specialized tissues. Then we get to use our next feature, which is symmetry. If you have bilateral symmetry, you're going to be from this point onward. So everyone from here to the right has bilateral symmetry, except it's not on this branch. So if we follow this branch down, here we have a trait radial symmetry, the radiata. That's going to be our cnidarians, our sea anemones, our jellyfish. So we've covered this trait here now. Let's talk about germ layers. So germ layers are types of tissues that form in early development, and you either have mesoderm or you don't have mesoderm, and we divide our germ layers by counting toward the center. So the prefix di is 2, so this is going to be 2 types of tissue. So it's a diploblast, even though we don't say di when we say 2, we say di, but we pronounce diploblast. You can say diploblast. That's totally cool. I'm going to say diploblast. It's fine. So here I have my outer layer. We're going to use the prefix for outer, ecto. derm referring to the tissue. So I got my ectoderm. Then I'm going to have a weird non-living layer, but it's not mesoderm. And then I'm going to have an inner layer called endoderm, and then I'll have my space. So there's really two tissues, hence di. Now I'm going to have three tissues, triploblast, but I'm going to say triploblast because it's easier for my brain to say. You can say it either way, but think tri for three. In a triploblast or a triploblast, you're going to have your ectoderm, that blue layer. Then you're going to have your mesoderm. So now we have a second layer. And then our third layer, our endoderm. So it's 2 or 3 layers of germ tissue as your organism is developing. Diploblasts or diploblasts, there are those 2 germ layers we just discussed. They have a simpler internal organization, and many of these things are going to be radial symmetrical, but we also have our flatworms in this group. And so I've shown you a planaria because I think they're adorable. And if you were to cut them open, they would have the endoderm and the ectoderm only with a thin layer of that non-living tissue. For the triploblasts, we're going to have those 3 layers. So you're including in mesoderm. This is going to allow you to develop muscles, organs, complex cavities. And this is true for most of our bilaterally symmetrical animals, such as this true worm, our oligochaete. This is an earthworm here. So if I were to cut this earthworm across and look in, it would have ectoderm, mesoderm, and endoderm. So it's got those 3 layers. Triploblast. Now that we have those different germ layers, we can make our bodies more or less complex. If we have a body cavity, we call it a coelom. I know this word doesn't look how it's pronounced. It looks like colomes, but it's pronounced coeloms. I'm not exactly sure why. If you want to say colum, that's okay. Remember, just say things with confidence and everyone will believe you. But I'm going to say coelom because that's how I was taught it was said. These are going to allow us to get more and more complex with our body type. So here we have an acoelomate. If we put A in front of it, it means they don't have it. So acoelomates, they don't have an interior body cavity. Instead, they have their ectoderm, they have their mesoderm, and they have their endoderm. Then they have a little hole which they run their digestive foods through. For example, here we have a really pretty flatworm. This is an acoelomate. Now we're going to talk about things with an actual coelom. If you put U in front of it, it means true. So this is a true coelomate. This is going to be pretty much everybody. We have annelids, which are worms, mollusks, arthropods, echinoderms, which are starfish or sea stars, and then chordates, which are us. So we have a true body cavity. Now let's go from the outside in. I'm going to have my ectoderm. I'm going to have my mesoderm, but now my mesoderm connects and it wraps around my endoderm. And in the process of doing that, I've created this sac, this space. That's a coelom. That's a body cavity. It's completely enclosed within the mesoderm. I can specialize in there. I can do things like build a liver. That's going to allow me to really have complex organs and organization. But that's the cool thing, is that it's fully wrapped in mesoderm. And in the center, I will still have a hole, which I will use for my tube of my digestive tract. So from mouth to anus is just a hole through you. It's continuous. You could swallow a string and fully let it go from the inside to the outside. That's that hole. Everything else that's not part of my digestive tract hole. So like think about your pancreas and your kidneys and all those things that are not part of it, that would be in your coelom. So that's where all of our cool organs lie. Here we have a picture of a worm, an annelid. Then we have a third option. So it does have something, but it's not a true coelom. And so it's a pseudo, which means false, a pseudo-coelomate. So that's false coelom. We have the ectoderm, we have the mesoderm, we have this weird space, but it's not surrounded in mesoderm. So because it's not surrounded in mesoderm, we're not going to get the complexity of organs. We're not going to get the complex organization. Following this weird space, the pseudocoelom, you're going to have endoderm, and then you're going to have your digestive tract. Examples, nematodes. These are roundworms. Nematodes are extremely common on Earth. They are everywhere. They're really important to us because they are a model organism. We've learned a lot of genetics by studying nematodes, but they do not have a space wrapped in mesoderm, and so it's not a true coelom, it's a pseudocoelom. Let's look at this trait on our tree. After we did our Hox genes, after we did specialized tissues, after we did symmetry, now in our bilatera, so all of our bilateral critters are going to be divided up based on whether or not they have a coelom, so these are eucoelomates, or whether or not they do not have a coelom, these are acoelomates. So acola, think coelom. That would be the group that does not have a coelom. We said earlier that when your gastrula gets that little push in, the blastopore, its fate helps us determine the classification of organisms. If that blastopore becomes a mouth, we say that you're a proterostome. I think proto first, the mouth was first. In a proterostome, the coelom often forms by splitting the mesoderm. This is referred to as schizocoely, which is just a fun word. And who does this? Arthropods, mollusks, annelids, which are our worms. In comparison, if your blastopore becomes the anus first, we call you a deuterostome. I remember this because duty, that just helps me think. So deuterostome, duty, the anus becomes first. In this case, the coelom forms by an outpocketing. We call that enterocoely. And this is going to include echinoderms, which are our sea stars, and then our chordates, which are us. Here we have a diagram. On the left, we have protostomes. Here's my gastrula. In my gastrula, I have my blastopore. I have that push in. That push in is going to become the mouth. As it travels through, the second hole created becomes the anus. So here is where my coelom will form. And then I have my right and left halves because these are all bilatera. In comparison, here's my deuterostomes, were deuterostomes. I have my blastopore, but note the coelom is not up here now. It's going to form from these outpockets. And the first hole is going to become the anus. As it pushes through and continues, the second hole will become the mouth. So mouth second. And the coelom will form on the right and the left sides, but it comes from these little out pockets. So the outcome is the same. The process is different. If we look at this on our phylogenetic tree, everybody with a body cavity is going to be either a protostome or a deuterostome. Most of them look like they're proterostomes, right? So in the protostomes, we have all of these critters. And in the deuterostome, we only have these two, but chordata is a complex group, and we like to think that they're extra special because we're in there. So here is our chordata and our echinodermata. Those are deuterostomes, which means the mouth formed second during the gastrulation phase of embryonic development. Now let's talk about metamorphosis and molting. So metamorphosis is where your critter goes from one body form to another body form, maybe doing a full, complete metamorphosis where it changes everything about itself into its final stage. If you do complete metamorphosis, your larval stage is often your feeding stage, and your last phase is your reproductive phase. So for a lot of butterflies, they don't even have mouths. They're done feeding. Their whole goal now is just to reproduce. Molting, on the other hand, is what defines a schizos. Say this to yourself. Ecdysozoa, which they will take their exoskeletons and rip them open and move out of them and do that periodically. And that's how they get bigger. I have a picture here of a cicada that is molting. So it's split its back and it is pulling itself out of its old exoskeleton, and it's now developing its new exoskeleton. The first couple days after doing this, they're really kind of sensitive, but then it hardens up and becomes a full exoskeleton. There's also incomplete metamorphosis. In incomplete metamorphosis, things like grasshoppers, they change body form and shape, but it's not so drastic as complete metamorphosis. So a baby grasshopper looks very much like an adult grasshopper, whereas a baby butterfly does not look anything like an adult butterfly. Check out this on our phylogenetic tree. These are the two groups that are going to do molting. We have our arthropods and our roundworms, and everybody else is going to go into this group here. So they're not doing molting, but there may be other changes in their body form. All right, now let's talk about the evolutionary history of animals. So our fossil record and evidence based on like volcanic activity shows that the animal kingdom didn't happen like once. There were repeated changes, there were mass extinctions, it was gradually reshaped, it changed many times. And so when we talk about our evolutionary history, we break it into eons, which are big periods of times, eras, which are smaller than eons, but are still big periods of time, and periods. Now, these periods help us break apart millions of years into smaller chunks, whereas an eon, check out how many millions of years this eon was. Just 100 or 540 is pretty amazing. Everything that happened before the Cambrian period, here's the Cambrian period, we call the, say it to yourself, pronounce every vowel, Ediacaran. So the Ediacaran period. This was 635 to 543 million years ago. And in this era, we think that animals developed from protists, and they looked kind of like coins or looked kind of like feathers. And we have a good amount of fossils from this era. They're named after the place that they were found in southern Australia, so these hills. And this is a newer name, which is why I'm calling it out. You might also note that on the old slide, it isn't even there. So this newer name is what we're going to be using now. So it's the Precambrian period. Here are those fossils I just discussed. This one is round. It looks like a coin to someone who described it. Here are your two more feather-shaped fossils, each of these millions of years old, and from that Ediacaran period. Our Cambrian period was from 542 to 488 million years ago. And this is a really exciting period because this is one of our most rapid animal evolutions periods. We say this is the Cambrian explosion because it just like exploded with diversity of animals. Most of the phyla that we're accustomed to today originated in the Cambrian period. For example, echinoderms, mollusks, worms, arthropods, and chordates all evolved in this period. We think that the Cambrian explosion has a couple different causes. There isn't really one, it's debated. We do know that there was rising oxygen levels. So here on this graph, we have millions of years before present. So this is right now, and this is 1,000 million years ago. Oxygen levels were really consistent, and then they started to increase, followed by a spike. Who makes oxygen? That's going to be autotrophs that are doing photosynthesis. As this was happening, ocean calcium levels were also changing. We had a lot of shallow seas, which had a lot of ecological variation that allowed for habitat differentiation. So critters that lived in shallow warm water versus tidal zones versus little pockets of deep cold water. We had a change in predator and prey relationships because if we're getting a lot more species, who is preying on you or who you're trying to eat will change how you live your life. So imagine I'm in this little tidal zone and there's a predator that always hangs out near the big rocks. Maybe I avoid the big rocks over evolutionary time. I could adjust to my new habitat, and that's how we get a lot of these different diversity of animals. This is also where we see our genetic innovations, such as the Hox gene. So now I have a body plan gene that I can reuse or delete as needed. And all of these ideas are used to explain the Cambrian explosion but we don't know which one's best. And we think it's probably a combination of the two. I mean, of the four. In the Cambrian explosion, we have all sorts of amazing critters, specifically trilobites and these wonderful feathered-shaped predators. We have the development of sensory organs to help these predators become better at catching prey. Pictures like this, you notice the sponges, are described as the fauna of Burgess Shale. Burgess Shale is an area, it's a rock formation in British Columbia, up in Canada. And there's just a huge amount of these fossils of all these critters together. And so we use it to reimagine what the full ecosystem probably looked like. We think there was a great amount of diversity and all sorts of predator-prey habitat differentiation. So you can find a lot of artists' rendering of the Burgess Shale because there's so many cool fossils to get creative with. In the Cambrian area, we also had trilobites. They were the most successful early animals. I have 4 fossils for you to show you their wide diversity. This should have been W. Wide diversity. Also, they had these exoskeletons that were easy to fossilize because they contained minerals such as calcite. And we think that they were all sorts of things. We think they were predators. We think they were scavengers. Some might have been filter feeders. They could have walked or they could have swam. But the diversity is incredible. Some were just millimeters big and some were several feet big. So not just whether or not they have projections on their exoskeletons. If you're interested in trilobites, some of the fossils are just stunningly beautiful. So I highly recommend a Google image search. Really incredible creatures. They were the Cambrian ones. Then, to help explain the diversity of animals that we now have on Earth, we have to talk about mass extinctions. We have 5 major mass extinctions so far. These are based on the fossil record. So we'll see a whole lot of animals and then we'll see like none. So that means that something happened, the animals died off, and it does repeat globally, it's not localized. In these areas, when you have a big die-off, it's actually ecologically kind of beneficial because what was taking up resources and taking up habitat is now gone. And so now you can have something new come in, you can have more diversification. Let's talk about this graph. Here I have extinction occurrences, and this is for marine genera. When they say genera, they mean genus. So that's what genera means, the number of genuses that went extinct. And then we have millions of years before present time. This is today, 0. And then we go 500, 600 is the most on our Earth. 550 would be the most on this graph. We have peaks and extinctions. And then note that these colors back here, we have peach, orange, pink, purple, and blue. Every major mass extinction denotes a change between boundaries in our evolutionary time. So we have Ind Ordovician, that's when this one changed into Devonian. Then we have when the Devonian ended, that was a mass extinction. There were still huge extinction events in these periods, but the denoting factor that changes from one evolutionary time to another get their own name. So here, this is the Permian, this is Ind Permian. Then we enter the Triassic, this is the Ind Triassic. Then we enter the Cretaceous, and this is the Ind Cretaceous. And each of these 5 mass extinctions is really interesting in its own right. But let's talk about the difference between mass extinction and background extinction. So background extinction are lower rates of extinction that happen all the time. If we go back to this graph and we put a trend line, this would be the background extinction rate. So just the trend of how often something goes extinct without it being a catastrophic event. The catastrophic events are what cause mass extinctions. So these are rapid and they are global. In background extinctions, these can be local and it can be driven regionally. Like if I had an earthquake or a single tsunami, those could cause background extinction. The ecosystem bounces back gradually. And in mass extinctions, this is a major environmental disruption. This could be that asteroid or that series of volcanic eruptions where it happened hundreds of times and totally changed the atmosphere. These are going to reset the ecosystem really quickly. And in response, we're going to open huge niches, which are habitats for animals to exploit. And then we get to see this rapid evolution of organisms in response to the new habitats, hence the Cambrian explosion. So all of those little organisms that survived now have fully new areas to exploit, and they're going to diversify. I put my favorite extinct organism here. Do y'all recognize it? It's the saber-toothed tiger. I think they're so cool. Their skulls are just wonderful. And I love the artist rendering of what they may have looked like. So the largest mass extinction was the Permian-Triassic. It's the one that changed from Permian to the Triassic era. So it's also called the End Permian. It's this one. Note how high it is on the graph compared to the other mass extinctions. It's often referred to as the Great Dying, and that was approximately 251.9 million years ago. We estimate that 95% of species died. 95% of life on Earth died. And that's wild. So now the 5% of the survivors, where their descendants, they had a completely open world to start exploiting. And we had a lot of different diversification after this event. This did cause the extinction of the trilobites and major reptilian groups. We also allowed dinosaurs to radiate. By radiate, we mean diversify and grow into different species. And this allowed for the diversification of land plants. And now that we have land plants, everybody else can start moving onto land. So it's a really exciting period evolutionarily. We think that this was caused by extensive climate change and/or volcanic activity. When volcanoes erupt, they release a lot of carbon dioxide. When we say climate change, we mean like there's going to be ice ages or hot periods, or like everything is shifting. Now, the next mass extinction that we love to talk about is the Cretaceous Paleozoic. Here we have in Cretaceous, and this mass extinction was only 66 million years ago. There was a large meteor impact in the Yucatan Peninsula, and that added to volcanic activity, created an atmosphere that caused extreme climate change. And we think that created the mass extinction. So it wasn't just the meteorite on its own. Plus, we didn't have the meteorite idea until 1980. And I love pointing out how recent that is because we're still learning. If we're basing a lot of this on the fossil record, we need to find the fossils before we can come up with these hypotheses. And so there's a lot of work to be done. If you're interested in fossils, this is a wonderful career opportunity for you because we have so much work to do to figure out the gaps in our knowledge. In this, a lot of plants, most of our large tetrapods, and our non-avian dinosaurs went extinct. And by non-avian dinosaurs, we mean everything except for the ancestor of birds, because birds are in the dinosaur group. In response, though, this allowed the diversification of mammals, which is us, and birds and flowering plants, all of which are really exciting and all of which are what we see today. So the result of this mass extinction created our habitat now. Cool. That is the end of our lection for overall what's an animal, how they get to exist. I hope you had a good time, and I will talk to you soon. Email me. Bye-bye.